Hey guys, welcome to Grovebio channel. In today's session, we're gonna see about 2008 DBT question paper, Part B, Section 4. Question number 67. When synthetic mRNA consisting of alternating A and C residues and it was translated in a bacterial extract, only one kind of polypeptide consisting of alternating threonine and histidine is formed. So, when the base sequence was changed, like adding one A at the front, three different polypeptides were formed: polyasparagine, polythreonine, and polyglutamine. Hence, we may conclude one codon of histidine is, and the options are this. See here, you can see two possibilities: ACA and CAC, right? And here you can see three possibilities: AAC, ACA, and CAA. So in these two, you can see ACA in common, right? So this ACA can be threonine. So here threonine is there, and here also threonine is there. And for histidine, other one possibility is other than ACA, CAC is other possibilities. So answer is codon for histidine is CAC. Okay, so you can refer to the codon chart as well. Question number 68. Hybrid dysgenesis is asymmetrical. It is induced by. These are the options given. So the answer is P male and M female crosses. So how come? Let us see the explanation. Lab M type female, which is crossed with P type male. So this M type female is old strain, okay, and this P type is recently found in Drosophila. Okay, this P type female has P type male has transposable element. What is transposable element? It is introns, that is jumping gene. What work it does? It just randomly jumps into any other gene and then it will change or stops the particular gene function. So what does it produce? It produces sterile female. Okay, sterile means what? Gonadal dysgenesis may be occurred so that the female is sterile. How come? The zygote will have chromosomal broken and germline cell apoptosis so that the X is infertile. So why the X becomes infertile? It has no piRNA. Okay, so when this is reversed, like lab M type male when crossed with P type female, what happens? It produces fertile female. But how? The P element is now present in maternal side, right? So the maternal Drosophila is protected by piRNA. How come? It is complementary to the P element. So piRNA is complementary to the P element. So what happens? This P element in the cytoplasm sequestered by the pico RNA. So the egg is now fertile, right? So this is the explanation. Now we can see like in diagrammatic form, lab M type female. This lab M type female is old strain. This P type male is found only in recent days. Okay, so when crossed, it produces sterile female because of gonads are broken due to the paternal, like male transposable element. Okay, so it becomes sterile female because of the P element present in the male. So the answer is P male when crossed with M female, it produces hybrid dysgenesis because P is inherited directly. When in case of female, this P element becomes silent due to the piRNA present in its X cytoplasm. Okay, so next one. Question number 69. During RNA polymerase II transcriptional initiation, phosphorylation of following factor is essential to commence transcription. So what are the options? These are the options. So answer is polymerase II CTD. like C terminal domain is denoted as CTD. Okay. So explanation. Eukaryotes of RNA P like 1, 2, 3. There are three types of RNAPs. Okay. Among that RNAP2, that is, this protein consists of 12 polypeptides, among which RPP1 is largest among them. So this heptad repeat containing CTD, like C terminal domain, which is responsible for synthesis of mRNA and non-coding RNA. This RPP1 enhances or modulates the transcriptional process. So, for the phosphorylation, this RPP1 is needed. Okay. So, RNAP2 transcription, it has initiation complex like TF, that is transcriptional factor 2ABDEFH. Okay, this complex. When crossed with uRNAP, that is unphosphorylated RNAP2, this becomes phosphorylated in after the initiation process. Like after initiation complex bound with unphosphorylated RNAP, what happens in the elongation process? SCR5 and SCR2 is phosphorylated in the C terminal domain using CDK9 and as well as CDK7. So after which the phosphorylation occurs in the C terminal domain, then only mature transcripts are formed. So only CTD is phosphorylated, not the transcription factors. Okay, the CTD of RNAPs get phosphorylated, right? So the answer is polymerase 2 CTD. I'm saying again, this transcriptional factors doesn't phosphorylate, only the RNAPs get phosphorylated. Okay, so the unphosphorylated RNAPs get converted into phosphorylated RNAPs in the elongation process. At which site? At serine 2 and serine 5 site. Okay, next question. Question number 70. Typical nucleosomal organization of a gene is not found in. Options are human liver nuclei, malarial parasite, human sperm, and neuron. The answer is human sperm. How come? Explanation. In cells, nucleosome packages DNA, right? Normal cell. But in sperm cell, nucleosome only retains specific genes like starting site of genes, okay, and also the regulated and development process gene. The gene which is responsible for sperm regulation and sperm development is retained in the nucleosome, but not other genes. Okay, so the chromatin in sperm organized by selective pressure on the base composition, okay, to facilitate the embryo's gene regulation. So only needed gene are present in the site, okay, in the human sperm. Okay, next question. Question number 71. A mutational event inserts bases in the beginning of the coding sequence of a gene. The highest chance of altered protein being functional when the number of bases inserted is. See, in codons, how many bases will be there? 3 bases. So when any one or two bases is added, it's going to change the entire codon now reading, right? When 3 bases added, what happens? Only one protein will be formed. In additional, okay, so the highest chance of altered protein being functional is 3. So answer is 3. Explanation is very easy, as adding 1 or 2 or 4 bases can change the entire sequence. So that protein will not function as the mother protein, right? So the codons are changed, but by adding 3 bases, only one codons get inserted, not base, only one codons get inserted. So answer is 3. Okay. You can refer to chart as well. See, here one codon will be there, here one codon will be there, here one codon will be there. The intersection of three codons, three bases will give one codon. Okay, one codon. So next one. Question number 72. In a temperature sensitive mutant bacteria, at non-permissive temperature, there are huge accumulation of Okazaki fragments. The bacteria are mutant for. See, the bacteria cells is sensitive for temperature. So, at non-permissive temperature, obviously, what happens? The DNA replication is inhibited, so that what formed? Okazaki fragments are formed. So the bacteria are mutant for. The answer is very obvious. Answer is DNA ligase. So what happens when DNA ligase is absent? Okazaki fragments will be there, right? So ligase only ligates the DNA. So explanation: DNA polymerases DNA. Topoisomerase avoids supercoiling. Okay, gyrase also removes the coiling and supercoiling. So then what happens? The DNA ligates only ligates the niche. Okay, the DNA ligase, what it will do? It will join the Okazaki fragments and it makes the DNA's part without niche. So this is the answer. So thank you. If you have any doubt, comment me in the comment section below. Subscribe will help me. So thank you. If is any doubt, comment me in the comment section below. Thank you.